Good morning, everyone. My name is Vurchas Aditya Tomar, and today we'll be talking about variation in cells. I also made a video in which we talked about cells, their history, and the cell theory. You can watch that video for more information. So, let's start. First, we need to know why are cells different from each other. Cells are different from each other because they vary in functions. So let's talk about variation in number of cells in different organisms. If an organism is unicellular, it means they have only one cell. That one cell performs all the functions like digestion, movement, respiration, etc. Some examples of unicellular organisms are amoeba, paramecium, bacteria, etc. Multicellular organisms have more than one cell. They have different cells for different functions. Most of the animals and plants are multicellular. Now, let's talk about variation in shapes. Cells vary in shape because they have different functions. Some examples are human cells, which are spherical in shape, smooth muscle cells, which are spindle-shaped, skeletal muscle cells, which are cylindrical in shape, neurons are elongated with projections, red blood cells are biconcave disc-shaped. There are many more shapes of cells. Now, let's talk about cells which have no definite shape, meaning they are irregular in shape. Amoeba is a protozoa. It has pseudopodia around its border to help it move, capture food, and engulf it. White blood cells are present in our blood. They show amoeboid movement and protect our body from foreign substances. Now, let's talk about variation in size. Cells cannot be seen by our naked eye. This means that we cannot directly see cells. Then how do we see cells? We need a microscope. Cells can be from a few micrometers to a few centimeters. Micrometer is such a small unit that 1 millimeter has 1000 micrometers. Bacteria can be ranged from 0.1 micrometer to 0.5 micrometers. Neuron is the longest cell in our human body. Ostrich egg cells are the largest cells in the world. Size of the cell does not decide the size of an organism. The size of cells in humans, rats, and elephants can be the same. The function which the cell does decides the size of the cell. How do we see a cell using a microscope? First, we need to know the types of microscope. There are two types of microscopes, compound microscope and electron microscope. Most of the cells can be seen through compound microscope, but cells which are too small can only be seen through electron microscope. Specimen is the cell, tissue or microbe we put on the stage of microscope to be observed. The specimen is placed on a glass slide. Then it is stained. This is called wet mounting. Now you'll be thinking, what is a stain? Stain is a specific type of dye which is absorbed by the specimen. It gives the specimen a particular color. Why do we need colors to see the cell? Most of the cells are colorless, meaning they have no color. So it would be really hard to differentiate the parts of the cells. That is why we use stain. It makes it easier to locate the different parts of the cells. Some examples of stains are methylene blue, saffronin, and crystal violet. Dry mounting is a type of mounting which does not require water. It is done to inanimate objects. Today, we learned unicellular and multicellular organisms, variation in shapes, different shaped cells, cells with no definite shape, variation in size, and how to use a microscope. Thank you for watching this video and have a nice day.